Okay, so hi everyone, my name is Monique Swanepoel and I will be presenting my work on using genetic data to infer the population status of the elusive Suni antelope. Now just to give you a little bit of background, the Suni antelope, also known as Mesotrochus muscatus, is a very small and shy little animal with an approximate size of 36 centimeters in height. Now this animal is also known for its habitat that it loves, specifically the sand forest as well as our enclosed woodlands. Now the reason why I'm showing out the habitat in this case is because of the reason that if we go and look into the study with regards to the Suni antelope and look at the population structure, for instance, and we are able to infer that they are highly fragmented as a population, we are also able to infer that it is highly likely that the sand forest might also be fragmented. And as you also might know, is that the forests are currently highly threatened. So we can add in a bigger picture rather than just adding to our small antelope. Now, just to give you an idea about the distribution with regards to this antelope, it starts round about at the top from Kenya all the way down to KZN in the eastern parts of Africa. As you will note, there's round about 4 to 6 different subspecies that occur in these regions. Unfortunately, all of these subspecies have been described according to morphological features, and no genetic information were taken into account when they were described. So why is it important? Well, a study has been done in the United States on a captive population where they were able to show that they had 2 different subspecies, as you could see here in blue and red, with 2 different chromosome types or numbers. For instance, 52, and the other subspecies with a chromosome number of 56. But what was of concern was the fact that there was a variety of diploid numbers that appeared between 52 and 56, as you can see here. Now, the cytotype variations 53, 55 is not a normal number. It shows that there's an extra chromosome present causing problems, especially when these 2 different subspecies hybridize with one another. Now, the problem with that is that this study showed that the offspring from these hybridization events caused a decrease in the fertility as well as an increase in the perinatal mortality. And this is a problem. Why? This is only done on a captive population. This had never been done with regards to the population in the wild. We have no knowledge with regards to that. We don't know if they are reproductively isolated or whether or not there are any differences currently present in the subspecies. So, in addition to this, we know on the IUCN list that they are ranked as, or said to be, least concern, and said that they have a population number of around about 365,000. Now, for me, I'm not pretty sure about that number because it was taken in 2008. Now, as you know, we're almost at 2018, so 10 years have passed and a lot of things could have changed during this time. For me, it has been a concern because we went to Tzanini, for instance, last year, and we went to locations where we sampled in 2008, and the localities were completely open. There were no dense habitats at certain patches where we previously located and sampled, showing there was a lot of disturbance in the habitat of the Suni. We weren't able to pick up any presence of Suni in those localities, which is of concern for us with regards to that. In addition to this, we do not know anything with regards to the cryptic species, their movement, the genetic diversity that's currently present, and that is for us very important because we would like to maximize and maintain the connectivity between these populations, because in such a manner we can increase the genetic diversity if it is necessary. So to get to the point, in my study, we are looking at 2 specific questions. We are looking at how genetically distinct are the different Suni subspecies, and then what is the degree of connectivity between these Suni populations across the fragmented range. So, as I said, we are geneticists, so how do we add to this? Well, firstly, by looking at a lot of number of samples, we are able to say maybe there's this and this and this number of species. we are able to determine the genetic diversity that's going on. We are allowed, or we can determine the gene flow that's taking place, as well as the social structure, by making use of molecular sexing markers, as well as number of unique genotypes that we pick up, but this I will explain a bit more in detail further on. So what did we go and do? We basically went into the field and we collected Suni dung pellet samples. Now that means that basically we did not make use of any normal samples like your tissue or your blood samples that geneticists usually use. The reason for that is because this animal is so small, we can't find it that easily to dart it or get a sample from them, and they stress quite a lot when you do catch them. So we chose to work with a less invasive method. So it did sound quite easy, right? Go in the field, collect some samples, but that is unfortunately not the case. We have the problem that there is two different small antelope that co-occurs in these habits, namely the Suni antelope as well as the red duiker. Now, why is that a problem? Well, basically, their dung looks exactly the same. So if I had to ask you which one is which, can anyone tell me what is a Suni, for instance, Suni dung from here? So to answer that question, the one on the left-hand side is our Suni dung, and the one is on the right is our red duiker. Now, in the field, it's very difficult to distinguish with that. So as a geneticist, we take these samples back to the lab, we do DNA extraction, we sequence this DNA, and we place it back into a database where we compare it against other sequences. And then in the end, it tells us, yes, you've got a Suni sample, or no, you have to go back and take another sample. So to go back to the sampling, we wanted to answer two different questions. So the first one was with regards to the different Suni subspecies. So what we basically did is we went and sampled took some samples in KZN, as well as some samples above the Zambezi as well as below, so that we have a coverage with regards to the different subspecies with regards to those questions that we want to answer. In addition to this, we also want to answer the second question. So we like to go to KZN, and we chose 4 different nature reserves, such as Pinda, Tzanini, Tembi, as well as Ndumu. In the case of Tzanini and Tembi, you will note that the samples were taken quite far apart from one another. The reason for this was so that we can collect as many possible different individuals. And as you can see here, the red dots are actually already identified as red duiker individuals, so they are present. But in addition to this, we also went to Ndumu and we collected some samples. So with the case of Tzanini, we actually had previous GPS coordinates, so we basically went in the field where they were present. But in the case with Ndumu, we had no such information present, so we basically had to go on word of mouth of where they actually saw a Suni and walk into the field. But for interest's sake, for Pinda, this is a complete different case. As you can see, it's a lot more samples that were collected. Now, the reason for that is because we've got someone that's basically in the field collecting every single day, going to the same locations and picking up the freshest samples as they can. And the reason why we're doing this is because this can add a lot more of information with regards to the behavior of the Suni antelope. How do they move between sand forest patches? How many males and females are in patches? Those type of questions that we don't have any answers yet to, so we can add a lot of value to that. So with regards to genetics, what have we done thus far? Well, thus far, we have been able to show, so sorry, this is a lot of information, but you can only focus me on the final genetic tree that's at the bottom. And what's important here is that we have already been able to show that the Mushatas subspecies, if we compare it to the red one, which is Zuliensis, and the yellow one, which is Livingstonian, separately, we could pick up already a difference between 4.4 and 4.9% of differences taking place. Whereas if you compared only the yellow and the red subspecies with one another, we only picked up 2.3 differences, percentage of differences. That already shows us that something is going on with regards to that one subspecies, making you wonder, shouldn't we start thinking and opening our minds about the status of the Suni? Shouldn't we be starting and looking at the different subspecies and maybe making them different evolutionary significant units and giving them a different rank. In addition to this, we have also been able to go and look at the sexing markers, as I previously mentioned. So just to give an idea, this is just peaks that are showed here, and the one, if we only pick up one peak with that sample at that specific location, we know that it is a female. Whereas in the case, if we pick up two, we can know it's a male. But how is that really important? Well, if we look at two different patches, for instance, on our GPS coordinates, and we see a male was present in this one patch and another male was present in another patch, we can answer, okay, if this is the same individual, we can track back how it was moving between patches and how it's utilizing its habitat. In the case, if it was two different individuals, we are able to say, okay, how many males are maybe around one another, how far do they move from one another, do they move together, do they cross paths, those type of questions. But more particularly, the work that we've been doing in PINDA, those number of samples that we have can answer those type of questions more particularly. But how do I know which one, or which individual is which? Now this we do with the use of genotyping. So we have around about 16 microsatellite markers that we make use of. For said with that, I've only shown you 8 out of the 16, so don't stress too much, it looks like a lot of information. But all that I want to point out here is in the top row, these things work, these are markers, and then on the left-hand side is our individuals. Now what's important here is that with each marker you get 2 different alleles. And if you look at all the markers, you can create different combinations with regards to what alleles they carried in each marker. And each variation that we pick up, which is shown here in different colors, is the different genotypes that we pick up. That makes that individual, like our fingerprints, that makes that individual unique. And with that, we are able to identify different individuals. We are able to identify the movement and the connectivity around their habitat. We can also look at the genetic diversity that's taking place with regards to each nature reserve. We are also able, like I said, the number of individuals, especially with PINDA, we might be able to answer that question for them with regards to that. As well as we will be able to look at the population structure that's taking place. And in a bigger picture, how does this add to, just to conclude, is if we are able to look at the genetic information, we can add it to another study that's been taking place in PINDA, where they try to estimate the number of Suni in their habitat at this moment with the use of camera traps and with the use of dung counts. And if we combine this information and put it together, we can maybe answer bigger questions, and this information that we gather, we can add to other nature reserves, such as Tembi and Sanini, to add to their nature reserve as well in value, so that they know what to do with their management plans. Can they do camera trapping in different manners, or the sampling in different manners? So, in short, the genetic work that we've been really doing, we are really excited about what we are picking up currently. It's looking really, really good. Hopefully next year we'll come back with bigger results. But with that, I would just like to say thank you very much. This one is not working.